So as Sarah said, I'm Cara. I'm a scientist in the research team here at B+LNZ. And the research team sits within Farming Excellence, which genetics is also a part of. And as part of my role at B+LNZ, I focus on animal health. So as Sarah said, facial eczema and internal parasites. So I'll be going over what we're doing in that space. But first of all, I'll just run through at high level what the research team actually does. So we identify research needs in partnership with farmers, and this is through UMR surveys, farmer councils, through the Farmer Council Conference, or talking to extension managers, and also through our Farmer Research Advisory Group, and then ensure these needs align with the B+LNZ New Zealand strategy. We design, lead and manage research projects, so we do take them right from the technical detail where we're quite involved to managing the tracking of the project itself. And we work with other scientists to deliver this research. And we want really robust peer-reviewed science so that we are giving the best to our farmers. And one of the most important parts of the process is actually being able to communicate these findings to the farmer to improve uptake and actually make a real difference. So these are some of the things we are thinking about when we initiate a program and also right throughout. So is this the best science we can do? So with any problem that we're trying to solve, we take a look at the literature, see what's been done both nationally and internationally, and so we can build on that knowledge rather than just repeating what's already been done. And then also looking at other technologies, just to see if there's any other way we could actually solve this problem. We're also thinking about, will the research make a difference? Because ultimately that's what we want to invest in. And obviously, so can our farmers use the research? And this is really important. So we undertake research in a wide range of areas, obviously genetics and animal health, as I've talked about, but also all of these areas. For example, with greenhouse gas mitigations, we work with the Pastoral Greenhouse Gas Research Consortium to find ways to mitigate methane. And so I'll just be talking about a couple of areas now, facial eczema and internal parasites and what we're doing in this area. So this is just a very potted history of the facial eczema science in New Zealand, and this is just the significant events. So there's obviously a lot of other research that's gone into this area, but these are the main events. So facial eczema is not a new disease to New Zealand, been here for over 100 years. In the 1950s, we found out that it was attributed to a toxin-producing spore of a fungus, and this fungus is right throughout New Zealand and in the pasture. So a lot of work at that stage went into being able to quantify the number of spores and some management tools as well. In the 1980s, genetics got involved and Fe tolerance was found to be heritable, and then these programs were initiated. So why are we investing in facial eczema research? So it is still a top animal health priority for our farmers. When we do surveys, this always comes out as number 1 or 2, so it is important. There are animal welfare implications. There are limited treatment options available for farmers, and the selection for Fe tolerance requires an invasive test, and I will talk about this soon and what we're doing in this space. There's huge impact on farmer well-being, obviously seeing the stress of the animals suffering in conjunction with having limited management tools to actually address this. And it is a large cost to the industry. So it's been estimated up to $200 million in severe years, and this is just based on clinical signs of disease. So if you added in the impact of subclinical disease, then this figure is likely to be a lot higher. So why do we need new science? Most of the research is over 50 years old, particularly in those original management tools, apart from genetics. And with new technologies over the past 50 years, we think there can be more accurate and effective tools for farmers. There are also some emerging risks with the current tools that are available. For example, zinc oxide is going to be banned in the EU from next year as a treatment for pigs. So we just need to be aware of these things and have other options available for farmers. Also with the climate warming and the climate change, the likelihood of the fungus increasing its distribution and therefore FE spreading is likely. So again, we just need to be aware of these potential risks. And ultimately, FE still hasn't gone away. It's still a major problem for farmers. So what we're doing in collaboration with Ag Research is undertaking a research program proposal. And this does have the ambitious goal of eliminating facial eczema as a disease of livestock. So as I said, we're in partnership with Ag Research on this, and also scientists from Manaaki Whenua, and we have developed a program proposal based on the science knowledge gaps that exist and where information needs to be identified so that new tools can be provided to farmers. So for the last couple of months, we have been discussing this proposal with industry partners. So that because this is such an ambitious goal, we do need a sector-wide approach and everyone to be on board. So it's really exciting because there's been a lot of support and interest through these engagements, and just to find solutions and knowing that they still need to be found. So we're looking to submit this proposal to MPI for co-funding, and this is definitely still in proposal stage, so it is a long process. So we're looking at a large area. The focus of this program is very broad. So it goes right from the fungus and the toxin itself through to the pasture, so identifying management tools for the pasture and also new tools for the animal. Also, the economic and social impact on farmers will be a large part of this to really understand how much of an impact it's having. And adoption and implementation will also be carried out throughout this program. So we really want farmers to be on this journey so that any tools that will be developed are practical and also they are adopted early. So because this program will take a while until we know if it's successful, the first steps we are investing in is looking into an alternative test for FE tolerance. As I'm sure you're all aware, the current test involves dosing rams with a known amount of toxin and then assessing the effects so that those rams can be, the FE tolerant rams can be sold to commercial farmers. So there are obviously issues with this in terms of animal welfare implications of dosing animals with a toxin. It is not readily accepted by all farmers. It is the only test that's available. And so with an alternative test that may be more acceptable, then the genetic gain may be more advanced. There's also potential consumer concerns about having an invasive test like this. So we're looking at new science to avoid the need for invasive animal testing. And Axel Heiser at Ag Research is leading this program. So what do we need in a new test? So we want something that's minimal impact on the animal. So we are looking at blood, saliva, and fibroblast cells from an ear punch. We obviously want it to distinguish between tolerant and non-tolerant animals so that those genetic selection decisions can be made. We need it to be reliable and cost-effective and also suitable for a commercial laboratory. So by the end of this project, we want to be collaborating with commercial laboratories so that it is available for farmers. So there are 4 phases of this project, and we're taking a phased approach so that at each stage we can stop and assess the science to make sure it is still worth investing in. And we can be quite critical of it. So phase 1 has been completed, which was to establish the cells in the laboratory initially So as I said, that we're testing blood cells, saliva, and fibroblasts, growing them up in the laboratory, and then exposing those cells to the toxin and not, and then assessing the cells that are exposed to toxin and not for certain biomarkers, so using genes, proteins, and metabolites. This phase was successful, and there were biomarkers that were found between the toxic cells and not, so it was looking promising to progress into phase 2, which is exciting. And phase 2 is testing animals that are known to be susceptible or tolerant, and then looking at those biomarkers again to actually narrow it down and identify something for a test. So assuming that will be successful, we would move on to phase 3, which is developing it into a high throughput, low cost test. And then finally, a validation process, which will be testing a large number of animals on the procedure. So if this is all successful, we'd be looking at a test available mid-2023. Okay, onto internal parasites. So we've invested in internal parasites kind of continuously because it is such a big issue as well for farmers. And with drench resistance increasing, it's becoming even more so. So I'm just going to touch on a couple of projects that have recently finished and then a couple that are still ongoing. So these projects were led by Dave Lethwick at Ag Research. The first one was looking at the impact of pre-lamb drenching with a long-acting product on ewe growth, body condition score, and fecundity. And we also took a look at the microbiome of the gut on animals that were drenched with long-acting product and not. And what was found here is that there's unlikely to be a financial benefit resulting from administering a long-acting drench product pre-lambing. And this is important just for farmers to understand when they're making parasite management decisions, any risks or benefits associated, especially because long-acting drench products are known to give a higher risk of drench resistance. And the microbiome, there were changes between ones that were drenched and not, but what these changes mean is unknown. It was the first study of its kind, so further work would have to be done to actually understand what that actually means. The next project was in cattle and looking at the effectiveness of ivermectin drench actually getting to the parasites in the gut by 3 different application methods. So oral drenching, pour-on, and injection was used. And it was found that the application method depended on the parasites they were actually targeting. So most parasites were, the application method that was the most effective was oral drench. But for Osteotargia, it was actually injection. So again, this is just important information for vets and farmers to know when making parasite management decisions. On to one of the projects that is still going on at the moment is a case study in the Wairarapa at a finishing farm that has triple drench resistance. So this was comparing the introduction of refugia of lambs that were coming onto the property and comparing it with quarantine drenching, which is the normal protocol, and to see if it improved triple drench resistance status over time. So this was a group of breeders and farmers working together. There's about 25 participants, I believe, and they all got their drench resistance status by doing a fecal leg count reduction test and then shared all their results. So everyone knew exactly what they were buying in terms of parasites. And it's this whole idea of not taking a gamble, rather, you know what parasites you're actually bringing onto your property so you can make effective parasite management decisions and you don't have this wheel of parasites of a gamble you're taking. So early indications of this program suggest that it is improving the triple drench resistance status, but to actually confirm these results, we have to wait for a year or 2 to make sure that is still consistent. And the last project I'll talk about is creating impact through communication. So as I said earlier, one of the most important things for us is to be able to communicate the research findings. And so we're using parasite management as a case study for this to assess the impact of 5 different channels of communication. So we've got an animated video, a Drench Wise poster of drenching techniques, a podcast where 3 farmers talk about how they farm in the face of drench resistance, the Wormwise workshops, and also the Wormwise e-learning module. So we've engaged Scarlatti to help us out with this, and they have designed an impact model which compares all of these different communication channels to each other, and they've forecast change resulting from each communication method. And these are based on assumptions. And we're using surveys to inform this model and inform those assumptions. So the first survey we did was an initial online survey to see how far the campaign reached, and also to see what the baseline practice is of farmers. And then we'll do a follow-up survey next month, and this is to see if any of the communication methods used actually caused people to change their parasite management practice. So did it cause behavioural change? So the ultimate aim of this is to assess what communication channels supported change. So where should we be putting our investment in terms of communicating this research, so that we'll use it for other projects as well. And I believe there's a poll question coming up. We were just going to ask if anyone has seen these communication methods. Here we go. So have you actually seen any of these communication channels? And you can answer as many as you like. Oh, great. So yeah, there is still a few that haven't seen it, so that is good. Now you know they're there. And then we had one other poll question, which I think will be coming up shortly. So in the past 12 months, what have you done in relation to parasite management? So a lot have talked to their vet. That's great. Thank you for that. Okay, so the last slide. So ongoing, we'll be continuing with education and communication in terms of parasite management. An example of this is media releases that we put out when required. This one was put out jointly with Ag Research over the last month or so, and just talking about the importance of quarantine drench and how important it is with increasing drench resistance. And obviously the Wormwise workshops are also, there's quite a few of them on at the moment. So looking to the future, we are working on what the strategy looks like for the future needs of parasite management. With drenches failing more often, we need to understand this a bit better and see what research is required for farmers to be able to farm with minimal or no drench. We want to preserve the drenches we do have, so this is something we really need to consider and understand more. Also, whether we can create some kind of operational workflow with all the case studies and the research we've done to date, can we build that into a more cohesive package? And then also in the back of our minds is the potential for an alternative test for the fecal egg count reduction test. As I'm sure most of you know, it is a blunt tool, but it's the best we've got. So is there ways we can improve that? And just a note that all our projects that we're currently investing in and the ones that have recently finished up are on the research webpage, so you can check those out. It'll tell you a bit about the project and how much we're investing in them, and also contact people for the pieces of work, so you can always get in touch with them if you need more information. Thanks.
Awesome. Thanks, Cara. Always good to hear what the research crew are up to. Now, the first question's come through, and Dan has offered to help you out on this one, but I'll throw it to you first. Can you explain the fibroblast relevance to FE? My understanding is that they work to create proteins in the body, for example, collagen.
So it was really just an another tissue sample to trial, because it is something that is taken from the animal, and to see if any of those markers would be found within them. So the fibroblast, I didn't actually mention that, they didn't work as well as the blood and saliva, so at the moment we're not progressing with those anyway. But I don't know, Dan, you might have something else to add.
That was exactly what I would have said, Cara. Great answer.
Thank you.
Always good to have everyone on the same page. Next question, what is the best thing breeders can do to help research initiatives you have outlined to be successful?
It's a good question. I think it's that whole thing of just making sure we communicate. So I think, Sarah and I have talked a lot about just making sure that we are, just communicating about these kind of research projects more often. And then there will be chances as well to input into these programs. So I think it's just about keeping that communication open.
Yeah, great. And I guess on that line, can farmers get involved in the FE program?
Yes, definitely. So we will. So at the moment, the proposal is, I'm assuming they're meaning the big program. So at the moment, the proposal is with potential co-funders for review, and then we will be talking to the Farmer Research Advisory Group about it. And if we are successful, then we'll definitely be getting a wider reach to farmers to help us, just for their input and obviously samples. And then if there's any tools, we would definitely want them to be on board from day one.
Awesome. Yeah, that's great. In light of the alarming rise in drench resistance and the number of farms dealing with this problem, is there a plan to develop a Wormwise 2 dealing with resistance workshop?
I think I will throw this one to Dan.
Yeah.
Mr. Wormwise.
Yeah, absolutely, thanks. So I guess there's two things to that. So that's a good point, right? That's a Wormwise question as opposed to a beef and lamb question specifically. A Wormwise is just going through a strategy review just at the moment. Paul Crick and Robert Peacock and others were from the Farmer Council were involved in that process, and it has literally landed in my email box this afternoon, the sort of final shot of that. And that really will take, the goal there is it would really like to take Wormwise from doing what it's doing at the moment, which is having some impact, but perhaps not having as much impact as it could, and really turning it up. It was part of the offer we took around the around the referendum levy vote to go out to really put a lot of effort into improving our effort on Wormwise. So that's all part of that conversation, Kate, that was from, wasn't it? Yeah, so I couldn't answer right now, but that's in action as we speak.
Great, thanks, Dan. Is there any research planned on comparing wormfeck and Kala so either genetic trait can be used by breeders?
Not that I am aware of in our realm.
Might have Richard Shaw online, actually. He may be able to add to this.
I think that's more of an ag research question, or a genetics question rather than Cara.
I'm not sure if Richard's online, but
yeah, I'm here.
Thanks, Richard. Don't know if you've got any answer to that, but if you have, go for it.
Yeah, look, it's something I've actually just trying to do a little bit of pushing within ag research. I'm hoping our next round of internal funding we can get something extra going. We've shown some really good stuff in the deer industry, and let's just see whether, I know that there's more and more breeders looking for some better answers, and I think both tools have their usefulness, and it's just a matter of getting the right information. I think we're all being a little bit remiss in the last few years about looking for not so much what's the best tool, but what's the most effective tool for breeders and then ultimately for farmers to use. I think we've just missed that story a little bit.
Yeah, it's a good point. Thanks, Richard. Cara, another FE question. When the FE test project is finished, how will farmers be able to access it?
So it'll be through commercial laboratories. So in that final stage, we'll be working with commercial laboratories who are interested, basically so that it is up and running and ready by the end of the project. That's the goal.
Yeah, great. And one more FE question. At the FE Gold meeting in Napier, Russell proposed a research fee to add to FE dosing to help fund and support future research. Is this still needed? And how do we keep together as a coalition of the willing on improving FE tolerance options for the industry?
That might be, Dan might be able to answer that question in terms of a funding scheme, but
so that's first I've heard of that. I guess I wasn't at that FE Gold meeting there. In terms of funding at the moment from an industry point of view, we, Beef and Lamb has decided, and as we've been out through the referendum, as I said before, that this is a really important thing for us and we're going to invest levy funds in it. And as Cara and Axel are meeting with our industry partners and MPI, I'd like to think that we don't need other funding over and above that to help fund that research, right? This is what we're going to decide what we'd like to do, put some costs next to it and say, go and find the money to do it. The second question, how do we keep together a coalition of the willing? I'm really keen for that to carry on and happy for feedback, Annie and others, as to how we do that.
Yeah, great. Thanks, Dan. And I think that's all we have for you, Cara. Thank you very much for joining us.